So I want to discuss about some terminologies when we talk about functions of antibodies. So the first is opsonize. So opsonize means make a foreign cell more susceptible to phagocytosis. So the antibody covers this foreign cell so that it's kind of putting a sauce on the external material so that our own cell-eating cell, such as macrophage, recognize and can phagocytose. And agglutinate is the antibody is stuck together to form a mass for the external pathogens. So here's the diagram, protective mechanism of binding antibodies to antigen. So agglutination means that you can see antibody has a multiple valence. So it can grab double bacteria or multiple bacteria together, which enhances phagocytosis and also reduces the number of infectious units to be dealt with. And the second is opsonization by coating or putting a sauce onto antigen so that enhance phagocytosis by phagocyte cells. And antibody directly interacts to toxins or virus or bacterium so that it can neutralize that the bacteria and virus cannot affect or enter into our host cells. So blocks active sites of toxin. And the other is related to an innate immunity that we have in our blood plasma. There's a so-called complement system, which is tiny C1 to C9, which when it triggers, when it recognizes the bacteria is attached by antibody, then those fragments, constant fragments, multiple fragments are triggering our complement system. So they basically get into the bacterial cell wall and make a pore so that it ruptures and lyses the bacterium. So that's what antibody does to activate this complement system. And this lysis makes the internal content of bacteria to rupture and come out. So that it causes an inflammation. So these were triggers our immune cells to be activated and causes inflammation. And as the last one is the so-called antibody-dependent cell-mediated cytotoxicity, or we call it sometimes ADCC. What it means is some of the target cells, when antibody attaching to them, will actually cause our immune cells, our immune system recognize and kill the cell. So for example, we already heard about Herceptin, the anti-HER2 antibody as a treatment because some breast cancer cells have an overexpression of HER2, human epidermal growth factor receptor 2. And sometimes you want to understand how this HER2 antibody leads to the treatment or killing of breast cancer cells, which overexpresses HER2. So in some study, there shows antibody doesn't really reduces or block the HER2-based signaling, but still it is effective. So some studies suggest that this is maybe caused by this antibody-dependent cell cytotoxicity, because this HER2 antibody, such as Herceptin or trastuzumab, will attach to HER2 overexpressed receptors of breast cancer cells. Our immune cells recognize it and trigger cell-mediated cytotoxicity. So that could be a very important mechanism of antibody-based treatment, very specific treatment. So now let's summarize this. So antibodies bind to toxin or pathogens, so that preventing them from binding to receptors on our host cells and entering. So that we call it neutralizes. And another aspect is to activate effector cells. So what are effector cells? Effector cells are the cells that actively respond to a stimulus and effect some change, such as a plasma cell is an effector B cell, because once B cell is activated, the cell has to produce a lot of antibody, and those antibody producing cells are plasma cells. And there are effector T cells, that's an T cell that is actively responds to a stimulus, or master cell. So antibody binds to pathogens and link them together, and that we call agglutination and coat. And these coated antibody will stimulate our effector cells to recognize their fragment, which region, because antibody binds to these pathogen with the antigen binding site. The remaining part is constant region or constant fragment. So this is FC, constant fragment is recognized by the effector cells. So for example, phagocyte will phagocyte these cells, or master cell or neutrophils, and these cells will recognize it and degranulate. So it, and giving cytokines too. Also, natural killer cells release cytokines and cytotoxic molecules to kill those cells. So the final ones, as I mentioned, look at this cell membrane. There's a C1 to C9. A nice pore is made to lyse the cell or bacteria. So this is a function of antibody, because when antibody coats these external bacteria, it activates complement. The name comes from, it is complementary to our immune system. So these antigen antibody complexes activate this complement system, and that is part of our innate immune system, which means it's not that specific, okay? So it complements the ability of antibodies and phagocytes to clear the pathogens. So next time, we will study about how to manufacture monoclonal antibodies and monoclonal and polyclonal antibodies and how to make it. Thank you for your attention.